Hello, my name is Eden Udan Dunstan, and my matric number is 97977, and I am from Subgroup 7, Group 2.
Hi, my name is Asma and my metric number is 98267.
Hello, my name is Deji Aryan Anadridas with a metric number of 98698.
Hello, my name is Anailicia Anachimiam. My metric number is
There are
98766.
three objectives for this experiment. Firstly, to highlight the application of spectrophotometry in biological samples and molecules for the aim of identifying compounds for qualitative analysis and determining their amounts for quantitative analysis. Secondly, to determine the chemical properties of unknown compounds by evaluating their absorbance spectrum. Last but not least, to generate and analyze standard calibration curves for colorimetric methods of substance concentration. The materials used in the experiments are Bradford region, 1 mg per milliliter bovine serum albumin base A protein, standard unknown protein solutions A, B, and C, micropipettes, test tubes and test tubes rack, distilled water, pipette tips and its racks. Last but not least, cuvettes and its racks. First of all, 6 test tubes were labeled from 1 to 6. 4 milliliters of distilled water were added to each test tube using micropipettes. Then, 1 milliliter of Bradford reagent was added to every test tube. The SA protein standards of 50, 100, and 200 microliters were prepared in duplicate and added accordingly. One test tube containing no BSA was used as the blank for reference. After all additions, the contents of the test tubes were mixed thoroughly using a vortex shaker to ensure the proper blending. The mixtures from all of the test tubes were then transferred into the cuvettes. The cuvettes were then placed into a spectrophotometer to measure absorbance. The Bradford region responds to protein concentration by changing color, and the spectrophotometer, set at 595 nanometers, provided absorbance readings used to estimate the protein levels in each sample. For part 2 of the experiment, the procedure focused on determining the concentration of unknown protein samples A, B, and C. The steps followed were similar to those in part 1, with the only difference being the replacement of BSA protein with the unknown protein samples. The same volumes of reagents were used, such as, in this video, 1 milliliter for distilled water. Then, 4 milliliters of Bradford reagent was also added to all of the test tubes, as it helped in determining the concentration of proteins in solutions. 2 test tubes were prepared for each unknown protein sample, which included A, B, and C, making a total of 6 test tubes. 1 additional test tube was prepared to serve as the blank or reference. The test tubes were mixed thoroughly using a vortex mixer, one at a time, to ensure proper mixing. After all of the test tubes had been shaken by using the vortex, all of the solutions were then transferred into the cuvettes. Before measurement, the spectrophotometer was calibrated using a blank cuvette with no protein. Then cuvettes containing unknown protein samples A, B, and C were inserted for absorbance reading.
To wrap up our spectrometry experiment, let's quickly summarize the outcome of the result. In part 1, we observed a clear positive correlation between the volume of BSA protein. The concentration of BSA protein standard derived from volume 50 microliters to 200 microliters. As the volume increased, the absorbance reading at 595 nanometer increased, going from 0.56 up to 0.185. This directly supports the Beer-Lambert law's principle that absorbance is directly proportional to concentration.
In part 2, we successfully generated a highly linear standard calibration curve, described by the equation of y equals to 0.02 times by x, with an r-square value of 1. This excellent linearity indicates a strong, predictable relationship between protein concentration and absorbance within our tested range. Using this reliable curve that we had obtained, we then determined the concentration of our unknown samples of A, B, and C using the equation. Sample B had the highest concentration at 13.4 microgram per milliliter. These results directly address our objectives. Firstly, we applied spectrometry for quantitative analysis and qualitative analysis. Next, we determined the absorbance properties of the protein dicomplex. And lastly, we had successfully generated and used a standard curve to find unknown concentrations. In conclusion, this experiment effectively demonstrated that the Bradford method combined with spectrometry is a valid and reliable method for quantifying protein, thus confirming its practical application in biochemical analysis.
Hello everyone. Today I'm going to share the key skills and concepts we learned from our protein analysis experiment using spectrophotometry under STB 1083 Biochemistry. So for the first new skill that we've obtained is the application of Beer-Lambert's law. First, we applied the Beer-Lambert's law, which states that absorbance is directly proportional to the concentration of a solute in a solution, provided the path, length, and molar absorptivity remain constant. In our case, we use this principle to determine the concentration of protein by measuring the absorbance of protein dye complexes at a specific wavelength. This showed us how spectrophotometry can be used to quantify biological molecules in a solution such as proteins. Next is instrument calibration. We also learn how to calibrate a spectrophotometer using standard protein solutions. Calibration ensures that the instrument gives inaccurate reading. We prepared a series of known concentrations and measured their absorbance to generate a standard curve, which was crucial for determining the unknown concentrations in our samples. Third, we use of software for data analysis. Another skill we gained was using Microsoft Excel to plot absorbance against concentration. This allowed us to visualize the linear relationship predicted by Beer-Lambert's law and use the graph to determine unknown values through interpolation. It also improved our understanding of how data visualization helps us interpret experimental results with precision. And finally is the preparation of standards. Finally, we learned how to properly prepare standard solutions using micropipettes. This improved our pipetting accuracy and consistency, which is vital in biochemical experiments. Accurate preparation of standards is essential for both calibration and the overall reliability of spectrophotometric data.
What are the challenges we face and which part it is difficult to conduct and why? First, pipetting errors. Tiny volumes are often used, and even slight pipetting errors can cause significant variability in absorbance readings. Inconsistent mixing and sample homogeneity. Protein can aggregate or precipitate, especially if not handled or buffered correctly, leading to non-uniform absorbance. Cuvette handling, such as fingerprints, bubbles, or improper alignment, can cause light scattering or absorption errors, such as affecting precision. There are few suggestions for improvement, such as use appropriate blend and controls. Always use a region blend that contains everything except the protein to zero the spectrophotometer. Include standard proteins to create a calibration curve for quantification. Lastly, use proper buffer and pH and choose a buffer that does not absorb at the measurement wavelength. Maintain a consistent pH as it can affect protein structure and absorbance.
In addition to using appropriate blend controls and maintaining the correct buffer and pH, there are two more improvements we can apply to strengthen our experiment. First, repeating each absorbance reading helps minimize random errors. It allows us to detect any unusual values and calculate a more reliable average, especially when plotting the standard curve. The other point is ensure consistency in handling. Second, keeping things consistent, like using clean cuvettes, placing them in the same orientation, and following equal timing for all samples helps reduce variation and improve the accuracy of our results. By implementing these improvements, we can ensure our future experiments produce more precise and trustworthy data.